Okay, let me go ahead and make a correction of something I said earlier. When we talk about oxidation reduction reactions, the important thing to remember is that when something is being reduced, it means we're adding hydrogens to it, basically because we're adding electrons to it. When something's being oxidized, it means we're taking hydrogens away from it. So here, when we see this area here that says oxidation reaction produce 2 NADH, we're pulling hydrogens away from the citrate. Okay, what I misspoke, I said that we oxidize NAD plus to NADH, when in reality we added hydrogens to it. So what I should have said is we reduced NAD plus to NADH. So again, the important thing to remember is if I pull hydrogens away, I'm oxidizing something. If I add hydrogens to something, I am reducing it. So I hope that is made clear for you. Again, what are our counts here? Well, we started out with our acetyl. We added it to our oxaloacetate to get our citrate. We pulled hydrogen out. We pulled a carbon dioxide out. We pulled another hydrogen out. We pulled another carbon dioxide out. We formed some ATP. We pulled 2 more hydrogens out to form FADH2, and we also formed more NADH. Now again, we'll talk about these numbers. So the next thing, let's do a quick review of what's happened overall, and then we'll finally talk about the last step, the whole reason why we're using all these hydrogens in the form of NADH and FADH2. Okay, we see one of our original pictures that has what I like to break down into 4 steps. Some people leave the preparatory reaction out as a step, but since it does produce hydrogens, I like to include it. So again, we start out with glycolysis. What do we get from glycolysis? Well, we got 2 NADHs, so we got hydrogens out of the breakdown of glucose. We actually got 2 ATP, even though we had to start out with 2 ATP to get the process. We formed 4 ATP, we paid back 2, and now what do we get? Our end product of glycolysis is pyruvate, also called pyruvic acid. Now again, pyruvate or pyruvic acid can either go into the mitochondria and enter step 2, or it can stay in the cytoplasm and be converted into lactic acid. What determines whether it will or not is whether we have enough oxygen going into the mitochondria to keep the process going. If we don't have enough oxygen for whatever activity this is being available for, then the pyruvate stays out. We go into anaerobic respiration and we produce lactic acid. We often see this occur whenever we're exercising and we're pushing it really hard. Okay, but in this case, let's say we have enough oxygen to continue the process. So our pyruvate, which was our 3-carbon product, and of course there were 2 of them, went through the first membrane, through the inner membrane space, through the second membrane, into the matrix. Well, while it was doing all that, it was actually giving off hydrogens. We actually formed some more NADH, and we lost a carbon dioxide. That's why the pyruvate became the acetyl, which helped start the citric acid cycle, because we lost our carbon dioxide. Now what else we see here is we see our cycle that was going on where we're pulling out more NADH, and also this time we're pulling out some FADH2. But we also know that the 2 carbons that came in on the lactate were also pulled back out, so we see that we're losing some more carbon dioxide. Well, we also see that we made a little bit of ATP. We made 2 ATP here, and we made 2 ATP here. So for 1 glucose molecule, so far we have 4 ATP. But here's this next step that we're getting ready to go over. This is where our major payoff is. This is where we're going to get the majority of ATP and why having mitochondria is an advantage for us. Okay, here's our last step, which is actually called oxidative phosphorylation, even though it occurs at something called the electron transport chain, which is very descriptive of what's happening. But let's make sure we understand where we are at in this process. Notice I have a fold here, and we said this inner membrane has these little folds, and we call these folds cristae. So hopefully you can see our phospholipid bilayer membrane, which is representing the inside membrane here. Here I've got proteins embedded in it, and we know proteins are usually functional for something, and we're going to see what the proteins are that they're functional for. And so I see proteins embedded in phospholipid membranes. Now, one thing we do have to understand is, remember, there's an outer membrane, then there is an inner membrane, and between the outer membrane and the inner membrane is an area that we call inner membrane space. So if you can imagine, there's another phospholipid bilayer out here between the outside of the cell, or excuse me, outside of the mitochondria, and this fold, which would occur somewhere on these inner membrane cristae folds. So I have space here that has the ability to collect things because it is between two membranes. It's really important to understand that. All right, now, so here we're in the matrix, and what was just going on in the matrix was I was going through the Krebs cycle, and the Krebs cycle was producing two things along with the first two steps. What is it that all three of the first three steps produce is they all produce NADH, whereas the Krebs cycle also produces FADH2. Okay, what are we doing with the NADH that came from the NAD+? What are we doing with the FADH2, which come from the FAD, came from it? All right, now, so what we're doing is, notice the NADH is now going to give off the hydrogens. We know the hydrogens are made up of a proton and an electron. Okay, so this NADH is going to give off that hydrogen. The hydrogen then will kind of split. The electron will actually travel through the electron transport chain. The electron will actually be picked up. The electron will actually power this thing right here, which is a pump. And the electron will power this thing, which is a pump. And the electron will power this thing, which is a pump. This is an electron carrier. It picks up the electron from that pump, carries it to this electron, powers this pump. Then this electron transporter will pick up the electron and transport it to this pump. Now, what are we pumping? Well, we're pumping the other part of the hydrogen atom, which is a proton or hydrogen ion. So notice that electron and proton was basically that hydrogen atom. Now the electron is being transported through the electron transport system to power pumps. What are we pumping? Well, we're pumping the proton part of the hydrogen atom. And where are we pumping it? We're pumping it into this intermembrane space, which is literally between this inner membrane with the cristae folds and the outer membrane. Now, as we discussed, what happens if we get a high concentration of something on two sides of a membrane? Well, we know that by the membrane transport mechanism called diffusion, once we get a high enough concentration, it will want to run from the high concentration back to where it's lower concentrated. So when I bring the hydrogens here and I pump the proton part of it into the space, it builds up a concentration gradient. And then this concentration gradient will go from high to low, as all diffusion does. Okay, now, when it goes from here to here, it can't go through the phospholipid bilayer. It can't do simple diffusion. So it has to have some sort of doorway to allow it back in. Just so happens this doorway that allows it back in is actually our enzyme, our ATP synthase. And what that enzyme is there for, remember what enzymes do. They lower the energy of activation. They allow two things, in this case, two things to be put together. They allow the phosphates to be added to ADP so we can literally get ATP. Now, because we still need an energy source to actually cause that chemical combination to come together, the energy source is literally the movement of the proton back through the enzyme. That movement then is converted into this chemical energy of adding the phosphate back to ADP. We call this process of the protons moving through, energizing the enzyme, so now the phosphate can be added back to ADP. We call that chemiosmosis. Okay, now, again, remember what the enzyme does. It just basically has two active sites that will allow these two things to get very close together. But I still need the movement of the proton to energize that bonding of the phosphate back to ADP. And then once my ATP is formed, then it has the ability to leave and go be used as cellular energy. But we still haven't answered the question of where does oxygen come in? Why? We've talked about how we've actually use the hydrogens and how we've actually built up this osmotic or this diffusion gradient here. We've talked about how phosphate is added back, but we haven't talked about what oxygen's done. So notice, looking here in my diagram, I actually see oxygen right here because we know when we breathe it in, it's O2. But we know that each oxygen molecule actually has, or each oxygen atom, excuse me, actually has two openings in its valence shell that it can accept electrons. So once our electrons are going through the electron transport chain, they eventually get to this last protein pump. Excuse me, this protein pump that's pumping protons. Now I know that sounds a little confusing, but that's what it's doing. So where do the electrons, once they go from here to here to here to here to here, where do they go? Because if they're here, I can't pass any more through here until I empty the electrons that were basically transported to this area. So here's where oxygen comes in. The oxygen comes in and actually accepts the electrons from this pump. And once it accepts it, it also accepts some of these protons coming back in and is converted from oxygen to water. So what happens to the majority of oxygen that you breathe in? Well, it gets converted into what's called metabolic water. Now, that's not enough water to sustain you. You still need water from external sources, but it is a source of water for the cell, and that's what's actually happening. Now, what would happen then if, let's say, I'm being active and I need ATP for my activity, but I don't have enough oxygen to come in and accept all this? Well, what would happen then? If I can't get oxygen in here, then once the electrons got to this point, they would stop. That means this would stop, this would stop, this would stop, this would stop. My Krebs cycle would have nothing to feed the hydrogens to because my NADHs would, and my FADH2s would still actually, not have a place to deliver the hydrogens and the electrons to. So everything would back up. And then if we follow that logically, we know if this stage backs up, then it backs up the Krebs cycle. If the Krebs cycle backs up, then the preparatory reaction is not going to occur. And if the preparatory reaction doesn't occur, then we know that the pyruvate, instead of going into the mitochondria, would literally stay out in the cytoplasm. And that's when we would actually produce our lactic acid. Now, another nuance here, another thing I want to point out is notice that NADH usually starts out at this first cytochrome protein pump, but the FADH2 never starts at this first pump. It always starts at this carrier. So what that means then, as we're going to see here in a minute, is that NADH has the ability to actually power 3 pumps, and FADH2 only has the ability to power 2 pumps. So we literally get a little bit more out of the NADH than we do the FADH2. This brings us to one of our final PowerPoint slides that we need to understand, and you need to be able to interpret it. And so let's see what we've got here. We've got step 1, our glycolysis, which occurs in the cytoplasm. We're breaking glucose down into 2 pyruvic acid or pyruvate molecules, 3 carbons apiece. We see that we actually get 2 ATP from that. Even though we did start out with 2 ATP, we made 4, but then we paid back the 2, so we get a net gain of 2. What else did we get from this process? Well, notice here we actually got 2 NADHs, meaning we did pull hydrogens out of this process. Okay, now pyruvate, again, now we're out in the cytoplasm. If there is enough oxygen, and let me adjust this for a second. If there is enough oxygen, then we know this will proceed into the mitochondria, as we see here. So this is my step 2. This would be the transition reaction right here. Now notice I didn't get any ATP from the transition reaction. And notice I converted pyruvate, a 3-carbon molecule, 2 of them, into 2 2-carbon molecules. And notice to do that, I had to get rid of 2 carbon dioxides. So to go from here to here, I didn't get any ATP. I did release 2 carbon dioxides, but what else did I get? Well, I got 2 more NADHs. In other words, I pulled more hydrogen out of this conversion from pyruvate to the acetyl. Now, the acetyl then goes through the Krebs cycle, citric acid cycle. What do we get from that? Well, in one turn, we got 3 NADHs, but we know we had to go through 2 turns, so we got 6 NADHs.